small molecule drugs, because like as you point out, you can in most cases synthesize them with chemistry, just like basic like high school chemistry stuff. Test tubes, mixing things together, presence or absence of a catalyst, heat them up, cool them down, different things happen. Those processes tend to be extremely scalable.
Mhm.
And that's why, drugs like aspirin are very, very cheap,
Mhm.
particularly after they go generic and widely available. So aspirin's a good example. Antibiotics are another good example.
Mhm.
Now, biologic drugs, by contrast, are so complicated that we can't use those kind of building block type chemistry approaches.
Mhm.
The only way to manufacture those is in some kind of a living cell.
Got it.
And we have to hijack the existing machinery that nature gave us through genetic engineering to coax, in the case of Humira, it's a mammalian cell called a Chinese hamster ovary cell,
Okay.
into doing something you wouldn't normally do,
Sure.
which is to pump out a whole bunch of this monoclonal antibody protein that is the Humira drug. Hamster ovary cells, I mean, really, if they had their druthers, I think they'd prefer to grow inside a Chinese hamster's ovary,
Sure. Yeah.
right? So instead, we have to trick them into growing in a giant steel tank,
Mhm.
and that's not so easy.
Mhm.
In addition, the hamster ovary cells, they're evolved in that particular setting to make the proteins that a Chinese hamster ovary needs,
Mhm.
not human monoclonal antibody.
Uh-huh.
So we also have to trick it into that second trick. All of that requires an immense amount of effort.
Mhm.
And the tricks involved really explain why it is that over 100 years elapsed between when in the late 1800s, when Paul Ehrlich first coined the phrase magic bullets to describe antibody drugs,
Describe antibodies.
and the 1980s, when they got the first antibody drug into human clinical trials.
Mhm.
The capital expenditures, the cost of buying all of that gleaming stainless steel and all the piping and things, particularly on the downstream side, if you break down the cost of one dose
Mhm.
out of a biomanufacturing process, the capital expenditure to build the facility is typically 60 or 70% of, because you have to spread that over all the doses they make over the lifetime of the factory. That's how
Sure.
the accounting rules work. It's typically 60 to 70% of it is the equipment and the capex.
Mhm.
And so if you want to get, as our goal was to get the cost, per pure gram of protein therapeutic equivalent down by at least 100-fold,
Sure.
then you cannot do that unless the capital expenditures are dramatically lower
Right.
than on a traditional system.
Right.